We are going to discuss about the plant tissue culture. It is an overview part. Then I will proceed through the sections that has been given in your syllabus, especially for the biotech students of fifth semester. Now, what is plant tissue culture actually means? Plant tissue culture is a collection of experimental methods that will help us to grow large number of plants from isolated cells or plant tissues under sterile and controlled condition. Now this is very important, and also it is very advantageous since we can able to develop large number of plants which are endangered, and also the plants of medicinal importance and of commercial importance plants can also be developed from this technique. Nowadays, plant tissue culture is merged with the plant biotechnology part, the recombinant DNA technology, where we can genetically engineer the genome of the plants to give rise to of such type of plants of special importance, like for example the BT cotton that we know, which are such type of crops, such type of cotton plants that are have insecticidal activity. Okay, so let us briefly little bit discuss about the history of these plant tissue culture. About 250 years ago, that is in 1756, Henry Lewis have demonstrated callus formation on the decorticated regions of elm plants. From the elm plants, Henry Lewis have developed callus formation. Callus is actually an unorganized mass of cells which are parenchymatous in nature. That means it contains parenchyma cells tissue, and generally the callus tissue forms on the wounded part of the plants. But here, with the help of tissue culture techniques, Henry Lewis have tried to grow callus formation from that elm plants, from that decorticated regions of elm plants. Now, many botanists have regarded this work as the forward for discovery of plant tissue culture. Then comes the German botanist Gottlieb Haberland in 1902. He was regarded as the father of plant tissue culture because he have first developed the concept of in vitro culture. In vitro generally means within the lab. So he have made that concept that we can grow large number of plants within a short period of time and space, irrespective of seasonal variation, within the lab through plant tissue culture technique, where we will aseptically grow the plants free of disease, and we will grow the plants not in soil, but initially we will grow the plants in a mixture of chemical substances, just like the chemical medium that we prepare in case of, just like we have chemically cultured media in microbiology for the growth of bacterial cells. Similarly, here also we will use different type of mixture of components of chemicals called macro elements, micro elements, vitamins, amino acids. Okay, and also the phytohormones, the plant hormones like auxin and cytokinin, gibberellins can be used together to make a artificial medium, and from in that medium we will grow the plants for a certain period of time until it have achieved its root and shoot formation purely. Then we will then from that in vitro culture we will place them in the greenhouse, in a greenhouse for its stability, for its acclimatization, and we will then plant them in the field. So he have developed that concept that we can grow initially the plants within the culture. So much of trees are getting cut down by the due to urbanization, due to rapid industrialization, so we can make a backup of those plants also. And he was the first to culture the isolated and fully differentiated plant cells in a nutrient medium. Now that nutrient medium composed of many mixtures. You will see later on in the next videos that different scientists have prepared their special medium for the growth of special plants in vitro. Okay, like Murasij and Skog medium is the common medium which is normally employed for the growth of various type of dicotyledonous plants in aseptic condition. During 1934 to 1940, three scientists named Gautharet, White, and Novgorod have largely contributed to the developments made in the plant tissue culture. So Gautharet, White, Novgorod, they have also contributed for the improvements in the plant tissue culture. Well, good development has also been done by India as well. Two scientists named Maheswari and Guha in 1964 from India, they were first to develop anther culture. And what do you know by anther? You know that it is the male part of the plant and main reproduction part of the plant, which give rise to pollen grains. Okay, so they have demonstrated that we can able to use this anther and pollen culture for the growth of haploid plants, that means having half set of chromosomes. Okay, similarly ovule culture is also being done. It was actually prepared by some other scientists as well. So much improvement, like Stewart and Reinhardt in 1959, they first discovered somatic embryo production in vitro. These also look like strange that we can able to prepare embryo from the somatic cells of the plants with the help of combination of different hormones, phytohormones, the plant hormones like auxin, cytokinin, gibberellins. Okay, in a particular ratio to give rise to embryo from the somatic cell. So such type of improvements have been done in plant tissue culture. So I have said that what is plant tissue culture. Plant tissue culture is a collection of experimental methods that will helps us to grow large number of isolated cells or tissue under sterile and controlled condition. Okay, what type of sterilization is needed? Obviously it's a very aseptic condition. Okay, just like you have seen in microbiology that how we maintain the petri plates that contain the culture bacterial cells aseptically. Even a slight mistake can cause contamination from the air. Here also we maintain the plant tissue culture in a uh, very sterile condition. Okay, a minute mistakes can leads to destruction of your whole lab setup. Now, to carry out the experiments using tissue culture technique, well equipped laboratory is first required to have these things. Now this is one of the disadvantages also. It is very laborious, first point. Second point is it is very costly, because one time investment we have to be costly, because so much of things are required for the plant tissue culture. Like you need a washing area, separate room for washing area, hot air oven where within a certain temperature you will dry all the stuffs, all the things that are need to be carried out for tissue culture. Refrigerator for storing the sensitive chemicals which can get damaged at normal temperature. Distillation plants for the preparation of double distilled water, deionized water. Then weighing balance for obviously for weighing the chemicals needed for preparation of the tissue culture media that I am talking about, the artificial media. Then pH meter has to be maintained while preparation of the media. It has been said that 5.6 to 5.8 is the optimal pH that should be prepared while when you are preparing the tissue culture media, the culture media. From that media, in that media we will grow the plant tissues, right? Then we required vacuum pump. Vacuum pump is specially required for filtering the liquid media. If you are culturing single cells, okay, or small part of cells, not tissue, then we can use a liquid media known as and that type of culture is called suspension culture. Okay, because the cells are suspended in the liquid medium, and for that we need vacuum pump. Vacuum pump is required for filtering the liquid media, sugar solution through the filter apparatus using air suction. Then we require autoclave as well, because obviously we need a sterile environment. For this we need to autoclave. Now for autoclaving, what we will use, we normally maintain at 15 lb pressure, and temperature should be maintained at 121 degrees Celsius. I hope so. I will later on you will see that what is the accurate pressure and temperature needed, and it is maintained for 20 minutes actually. Normally in microbiology we maintain them at 15 minutes, okay, but here we will maintain them at 20 minutes, right? Then working tables, obviously it should be neat and clean, free from contaminants. Obviously the working table for the plant tissue culture will be separate from the microbiology, okay, otherwise contamination will occur. Heater is normally used for heating or warming the medium to dissolve the agar. Now, two types of medium we prepare. One is liquid culture medium for culture of the suspended cells, and we also prepare semi-solid medium that contains the agar. That same type of media that we normally do in microbiology also, but here more chemicals are being added, the macronutrients, the micronutrients, and so on. Now for melting the agar we need to heat the medium, and for that reason we need heater. We can also use microwave oven as well. Microscope for observation of the callus, okay. Wooden racks for storing your cultures after inoculation, proper inoculation of the cultures in the plant of the tissues in that medium. We have to incubate them properly, we have to check them, okay, throughout the period of time. Then comes the laminar airflow hood. Now this laminar airflow hood has to be different from the it should be separate from the microbiology laminar airflow hood. Otherwise, now if you are using the same laminar airflow hood, you have to then sterile them completely by putting switch on the ultraviolet light for minutes, for some minutes. Then we have to sterilize them completely with alcohol, with 95% alcohol, and so on and so forth. So laminar airflow hood is the best area for inoculating your tissues, the plant tissues, into the culture containing vessel or culture containing tube. Then culture room should be also be present. In the culture room, um, it should have controlled temperature. Temperature is maintained 27 plus 2 plus minus 2 degrees Celsius, not more than that, not less than that. Otherwise it can cause the growth of some other contaminants as well, and it can create problem. So this is the image of a hot air oven. Now you have already seen hot air oven, I hope so. uh, Hot air oven is normally used for drying the tubes, drying all the stuffs that we have washed them thoroughly with water, with chemicals, okay. Then comes, this is the autoclave, okay. Now this is a small autoclave. Generally large autoclaves or digital autoclaves are nowadays also used for sterilization, proper sterilization of the culture, okay, of the culture medium, as well as we also sterilize the all the things that are to be required during the inoculation process in the laminar airflow. Then this you all know the picture of laminar airflow, it look like this. Now there are different grades of laminar airflow, level 1, level 2, level 3, okay, depending on what type of culture you are going to do perform here, okay. Now, this is actually, I have said, the working principle of laminar airflow. You can study them. Now, laminar airflow has number of small blower motors. Actually, if you see the diagram, this laminar airflow have small number of motors, blower motors, that will blow the air, which passes through a number of filters, and that filter is called HEPA filter, high efficiency particulate air. I think you all of you know the name of HEPA filter, because nowadays we are trying to use the N95 mask that contain HEPA filter. HEPA filter is very very costly, and that's why the N95 mask is so much costly, the original N95 mask. Now this HEPA filter layer, about 2 to 3 layers, is present in this laminar airflow hood. And the laminar airflow hood, as you can see, the motors, the small blower motors, they are blowing the air, and that blow air is passed through that HEPA filter, that high efficiency particulate air filter. And such filters can remove the particles which are larger than zero point 0.3 micrometer. Okay, so those particles which are larger than zero point 0.3 micrometer are microbes, are bacteria, not virus. Okay, bacteria, then fungi. Okay, that can be skipped off, that can be trapped. Okay, allowing the air, ultra clean air which is free from fungal and bacterial contaminants, flow at a velocity of 27 plus minus 3 per minute, okay, through that working area. And two types of laminar airflow occurs, are there. One is horizontal laminar airflow, one is vertical laminar airflow. Depending on that, we use them. Normally vertical laminar airflow is normally employed for the students, for the research. Okay, but for commercial we can also use horizontal laminar airflow if number of people are working in a particular area in a large laminar airflow hood. Okay, now this is the culture laboratory that we can see. You can see so much of jars are containing, and each of these jars contain your the culture medium, and in that culture medium we have inoculated our plant tissue known as explant. Okay, and they are maintained, in a controlled culture conditions at plus minus at 25 degree plus minus 2 degrees Celsius. And there should be a humidity control also within this chamber, within this culture room, of 50%. Also more things to say, that this as they are plants, they need a normal phototropic period also. So 16 hours light period and 8 hours dark period is required, and that is maintained in this culture room. And that's why we use here light sources also. You can see tube lights are there. You can also use other source of lights as well, but there that has to be a capacity of 3000 lux light luminous capacity. Okay, not more than that. You cannot use halogen light or such warmer source of light. Okay, the normal light of 3000 lux luminous intensity, and that should be maintained for 16 hours, around the 24 hours. That means 16 hours light phase and 8 hours dark phase. And humidity is controlled at 50%. You can also try to maintain them at 40% as well. And temperature should be maintained 25 degree plus minus 2 degrees Celsius. Okay, now an ideal tissue culture laboratory should have a big room for general work, and small room for aseptic work, and for keeping the autoclaved articles. So the large room should be present for keeping, for your general work, all the sterilization process, then keeping or tissue culture work in within the laminar airflows, that should be done in the big room. And small room should be required for aseptic work and for keeping that autoclave particles. And the most important aspect of this in vitro technique is to carry out the whole operations under aseptic conditions. Bacteria, fungi are the most common contaminants in the plant tissue culture, and as they grow faster in the culture medium also. because the culture medium that we are using for growing that plant tissue, that same culture medium is very very good for growth of the bacteria and fungi as well. So it should be protected from them. And that's why we, when we are inoculating that explant, when we are inoculating that plant tissue into the culture medium, we thoroughly wash them. We do surface sterilization with the help of antibiotics, with mild detergents like Twin 20, okay, for a few minutes. Even we use a dangerous chemical as well, dilute solution of mercury chloride, just before inoculating. Okay. Laboratory for aseptic inoculation should be without any window or ventilator. Yes, this is needed. Normally, we know that in the laboratory there are working areas. There should be exhaust fans for removing away the smoke from that emerges from the chemicals. Otherwise, suffocation occurs. But in this condition, we need a air condition installment. But at the same time, we have also to has to be think that the windows ventilator should not be present, because from that source, from that area, from that through that contaminants may come, dust particles may come. Okay. The laminar airflow hood is the most suitable, convenient, and reliable instrument for aseptic work. Laminar airflow has a number of small blower motor that I have already mentioned with you, that will blow the air, through which the number of filters known as HEPA filters, high efficiency particulate air, remove the particles of size which are larger than zero point 0.3 micrometer. That means it can able to remove the fungus, the spores, and the bacteria over the working area, and thus clean air can comes um, when you are when you are inoculating them into the culture medium. Then comes the culture room. As I have said, also culture room for keeping the culture and under control temperature, light, and humidity. Light should be maintained, humidity should be maintained, temperature should be maintained. And it should be maintained, the temperature should be maintained 25 plus minus 2 degrees Celsius, that is from 20, 23 to 27 degrees Celsius you can maintain them, not more than that, not less than that. Relative humidity should be maintained above 50%. Although when we will go for greenhouse, that is just after this, when the plants have enough grown, all the roots and shoots have grown maturely in the plant tissue culture in the lab, when we will transfer them into greenhouse condition, within the greenhouse the humidity is maintained in about 40%, but here initially it is maintained at 50%. Special design racks are also installed in this room to keep the culture tubes, vessel, flasks in proper position. Normally, we tilt the tubes, flask in a particular position so that all the explants are getting exposed to the light. And the culture room also have a rotary shaker also for the culture of suspension culture. Now, if the cells are grown on liquid medium, and they have to rotate them, otherwise they will settle down and they will not get enough nutrients. For getting proper nutrients, they have to be properly aerated, properly agitated. And for agitation, we need we need shaker for the suspension culture, so that the cells can get equal amount of nutrients, so that they can able to grow into callus, and from that callus we can able to place them in a semi solid medium for the growth of the plantlets. Now there are some terms that has to be used in plant tissue culture. We have to see that terms, and let us see. First is your explant. What is explant? I have talked about explant two times, but I have not said the meaning. Explant is the excised or cut portion of your differentiated tissue or organ is regarded as explant. So explant is the cut portion of your plant. Okay. Now explant can be taken from any part of the plant body. You can take explant as leaf, as stem, as root, but some things has to be some things has to be noted while choosing the explant. Like for example, when we are choosing the explant, you have to be note carefully that it should be actively dividing cells. It should have actively dividing cells. That means we should take explant from the meristematic regions. In earlier class, in your junior classes, you have learned about the meristematic tissues. They are of three types: apical meristem, lateral meristem, intercalary meristem, right? Generally, explants are taken from the meristematic regions, which are mostly used for the tissue culture techniques, because the meristematic tissues like apical meristem derive from either shoot apex, that means from the shoot tip, or from the axillary bud. They show lesser degree of genetic variability. That means these tissues, these meristematic tissues don't show so much of genetic variation, because the cells of meristematic explant, they actively divide by normal mitosis, and the cells are maintained at diploid level. So at normal mitosis, the cells of meristematic explant are actively dividing, cells are maintained at diploid level. So by culturing the explants taken from the meristematic regions, we can get a true copy of the mother plant having the same genetic makeup. Second important thing that I have not mentioned here in these notes is that meristematic regions are actively dividing cells, so there is such less chance or less scope of entry of virus or entry of other bacterial contaminants into that area, because they will not able to maintain that at that site, because all the time the meristematic tissues are actively dividing. So there is a less chance of viral contamination or pathogen contamination in that meristematic regions. That's why most of the plant tissue culture, those who are doing plant tissue culture, they take the apical meristems or meristematic regions of the plants, so that we can get a totally disease free plant and a true to type mother plant. The next thing that I will discuss is about the callus. Callus is normally an unorganized mass of cells which are parenchymatous in nature. That means they are mostly parenchyma tissue. Within the callus tissue, a localized group of meristematic cells appear during the callus culture. So when we actually normally induce, we are forcing the uh, explants, when we are inoculating the explants into the culture medium, na, we are forcing that explants so that they can convert into callus culture, callus tissue, and that callus, that unorganized mass of cell, will have a meristematic cells that appear during the callus culture, and such meristematic cells will give rise to roots, will give rise to shoots, or both of the root and shoot, depending on the concentration of phytohormones used in the tissue culture medium. So, for the growth of roots and shoots, first initially we have to convert the explants into callus tissue. And for converting the explants into callus tissue, we need a ratio of phytohormones, the auxin, the cytokinin, gibberellins, in a fixed ratio into the culture medium, so that induction of callus can occur from the explant. And when and once the callus tissue occurred, within that callus tissue there will be some meristematic cells which can actively divide and give rise to roots and shoots, or both of them, depending on the concentration of the phytohormones also. Like when the concentration of cytokinin are high relative to auxin, I will give you the example of some auxins, some cytokinins, right? So when the concentration of cytokinin are high relative to auxin, shoots are induced. So if the concentration of cytokinin is too much high than auxin, then shoot will be induced. Similarly, Similarly, when the concentration of cytokinin are low than auxin, then roots will be induced in that explant. And at intermediate concentration, that means more or less concentration of auxin and cytokinin, it will give rise to callus. Now, these normally occurred in case of dicotyledonous plants. What about monocotyledonous plants? In case of monocotyledonous plants, okay, this is the figure. Wait for a minute. Okay, so this is the figure of normal plants that you normally studied in your junior classes. From class 7 you are seeing the flower, the fruit, the stem, leaf, the nodes and internodes. We have to be careful about the nodes and internode area. Now within the node there are axillary bud formation, and we can use that part, this nodal segment, for the growth of callus tissue, and from there we can grow into a new plant. Okay, we can also use the root tip. Normally root tip are especially used for some important chemical production which are called as secondary metabolites. Okay, now this is the flower which we normally see. These are the some segments of the flower, the male parts, the male reproductive parts, the stamen, which consist of anther and filament. The anther normally consists of pollen grains. From there also we can do plant tissue culture to give rise to haploid plants. Similarly, if you see the female portion of the flower consists of ovary and ovule, we can also able to culture this ovule for giving rise to haploid plants as well. Now this has been demonstrated, this has been given also in your syllabus about the haploid culture. Okay, then I will study them. Now this is the normal way of how callus formation can be done, and from the callus how shoot induction and root induction can be carried out. So it has been shown that if there is intermediate concentration of auxin and cytokinin, then callus formation will occur. High concentration of cytokinin and low concentration of auxin will give rise to shoots, and low concentration of cytokinin, high concentration of auxin will give rise to root formation. Now this is normally done in case of dicotyledonous plants. Okay, what about monocotyledonous plants? Now, in general, monocotyledonous plants do not show pronounced response to cytokinin, and they need high concentration of auxins like 2,4-dichlorophenoxyacetic acid is auxin that can be employed at high concentration for induction of shoots and roots in case of monocotyledonous plants. You can also use abscisic acid and gibberellins as well for induction of roots and shoots in monocotyledonous plant. Well, I have said that I will say about some of the examples of auxin and cytokinin. One of the auxin I have already said, 2,4-D is the auxin, 2,4-dichlorophenoxyacetic acid. Others are IAA. Okay, let me write them down. IAA. IAA is indole acetic acid. Then your other are also there. IBA, indole butyric acid. Indole butyric acid. Then naphthalene acetic acid, also called as alpha naphthalene acetic acid. Okay, alpha naphthalene acetic acid as well. Okay, so these are the some examples of auxin. The examples of cytokinin are six. Examples of cytokinins are kinetin. Okay, examples of cytokinin is kinetin. Kinetin. Okay, the full chemical name of kinetin is your 6-furfuryl aminopurine. Then other names are also there like 6-BAP, 6-BAP, that is 6-benzyl aminopurine, zeatin. Okay, zeatin, zeatin, zeatin. Zeatin is also an example of a cytokinin, and 2-IPA, that is 2-isopentyl adenine. Okay, so some of the examples I have said about the auxin and cytokinin. Okay, let us move to the next term that has to be mentioned here: de-differentiation. What is de-differentiation? The phenomenon of mature cells reverting to meristematic state to produce callus is called de-differentiation. So what is de-differentiation? When the mature cells, they revert to form meristematic tissue, so that we can grow into callus, that is called as de-differentiation. De-differentiation normally done when the explants will give rise to callus tissue. De-differentiation is possible because non-dividing quiescent cells of the explant, when grow in suitable culture medium, revert into meristematic cell. It has been shown in the laboratory by the scientist that the non-dividing quiescent cells, quiescent means inactive, but non-dividing quiescent cells, they, when they are cultured in the tissue culture medium or suitable culture medium, these non-dividing quiescent cells, the inactive cells, can reverse to form meristematic tissue. There is a property called totipotency that I will discuss here. Totipotency is the ability of the each plant cell that can develop into a whole plant. Now, this property is present in our body, especially in the stem cell, right? The cloning process is being done by the use of this stem cell technique, but not all of our cells have the ability to give rise to a whole human being. But plant tissue, a plant cell is such type of cell that from any plant cell you can give rise to a whole new plant. That is called the protipotency property. So the ability of the individual cell to develop into a whole plant, that is referred to as cellular totipotency. The inherent characteristic features of plant cells, namely de-differentiation and the redifferentiation, that I will just now I will talk to you now, are responsible for this phenomenon of totipotency. So totipotency, or those cells which are totipotent, they must have these two characteristics. One is de-differentiation, where where the mature cells which are non-dividing, they reverse into meristematic tissue. And they can also go for redifferentiation property, where the callus cell differentiated into plant parts, into plant organs. So such strange type of, I don't say strange, but this is a special property of the plant cells, that they can able to revert into meristematic tissue if they are grown on culture condition. And then that callus that contain meristematic cells, they again they grow and differentiated into different plant organs, giving rise to a whole new plant. So both these two properties has to be present in order to become totipotent. We, in our stem cells, we are pluripotent, not totipotent. Okay, we have a totipotent property, but in case of embryonic stem cells, the ES cells, if you study in books, right? Now, this is actually about the overview of plant tissue culture, and some of them you have to study in your syllabus. Okay, some of them you have to study in your syllabus, you will see. Okay, wait for a minute. Okay, so you see that these plant tissue culture are of many techniques, many types, like callus culture, organ culture, where we culture the special organs of the plants like embryo, seed, anther, ovary, meristem, nucellus. Then comes the cell culture. Individual cells are cultured in liquid medium. Protoplast culture, where the plant cell wall is removed, and we cultured those plant cells, okay, in aseptic condition. Now we normally do protoplast culture for hybridization, for somatic hybridization, because we know that if we can able to remove the cell wall of the plant cell, the remaining portion of the protoplast can be used to fuse with the other protoplast as well, giving rise to hybrid cell. That is the somatic hybridization concept. And then we again revert back them to generate that cell wall, and we grow them, we culture them in liquid medium for the purpose of callus formation, and that callus formation, from there we do the callus culture. Now in callus culture it is done on semi solid medium, and this involves the culture of differentiated tissue from the explant, which will do the de-differentiation process in vitro to form callus. So they have to undergo that de-differentiation property so that they can form callus tissue. And from that callus tissue we will when we do subculture of that callus tissue in a certain medium that contain high concentration of cytokine in respect to auxin, so it will be induced. Another subculture where we will induce high amount of auxin respect to cytokine in root will be induced. In this way we will in a timely way we will grow the whole plant. The organ culture is the culture of isolated plant organs, referred to as organ culture. Separate organs of the plants are cultured, like we can culture the only the embryo, only the seed, root, endosperm, ovary, meristem, anther, nucellus can culture separately. Now, this callus culture and organ culture generally can be grown on semi-solid medium that contains certain percentage of agar to grow. Now, there are different types of organ culture as well, like there is an organized organ culture and unorganized organ culture. We will see them when I will discuss elaborately that how organ culture is being done. So, when a well organized culture of a plant, like your seed or embryo, is used in the culture, it is called as organized organ culture. So, if you are using seed or embryo as a part of organ culture, it is known as organized organ culture. And in this type of culture, the characteristic individual organ structure is maintained, and the progeny, the baby plant which will be formed, is similar in structure as that of the mother, as that of the original organ. In case of unorganized organ culture, this involves the isolation of cells or tissues of part of an organ and culture them in vitro, okay, to form callus, and from that callus we will grow the plants. In case of cell culture, individual cells are being used. They are observed in microscopically. They are obtained from the explant source or callus source, and these culture are carried out in dispersion medium. That means the medium containing liquid culture. So proper agitation should be provided, and that will give rise to small viable callus, and from that callus we will again do the callus culture. In case of protoplast culture, I have already mentioned that in protoplast culture we remove the cell wall of the plant cells. Now obviously you have a question that if we remove the cell wall, then it will not able to maintain that pressure, that balance pressure inside the environment of the protoplast, so it will get burst off. But no, we use certain chemicals of sorbitol mannitol solution to maintain that osmolarity at certain concentration, and then after maintaining that protoplast we do protoplast fusion or somatic hybridization, so that we can get a hybrid cell, and that hybrid cell is again reverted back its cell wall again. From there we will do cell culture, and from that cell culture we will develop callus in the liquid medium, and from that callus we will do the callus culture. Such long procedure is there in this protoplast culture. Now I given just an overview of steps of plant tissue culture. The same steps we will repeated and repeated when you will see the micropropagation technique. Okay, just I am giving the overview. You have to write the details also. So first is your steps of plant tissue culture is your preparation of instruments and nutrient culture medium. So we have to prepare all the things that has to be used in the tissue culture, like the instruments, the nutrient culture medium. Okay, now how it is being carried out, the all the things, all the stuffs that are used for the tissue culture, like your forceps, scissors, scalpels, blade holder, surgical blades, petri plates, having two filter papers, Whiteman filter paper, beaker having a 500 ml capacity, Erlenmeyer flask, test tubes, disposable pipette tips, bottles should be, these things are employed in the tissue culture, so these things has to be maintained, these things has to be kept in that place. Okay, and there has to be aseptic environment, so we have to wash all the stuffs with detergent with mild acid to remove away the dirt and other contaminants, because previously we have used for other things, for other experiments before, so you have to wash them thoroughly. Then we have to dry them in the hot air oven, the hot air oven that I have shown you the diagram before little bit ago, 60 to 80 degrees Celsius, but generally we use 60 degrees Celsius, not more than that. Then wrap in brown paper and we do autoclave at 120 degrees Celsius with a pressure of 15 Pascal for 20 minutes. Okay, so this is the preparation of your instruments. For the preparation of the culture medium, we use different type of chemicals as well. I have not shown you all the chemicals, a big list of chemicals are there, but for simplicity I have written macronutrients. Macronutrients are such nutrients which has to give in high concentration, because these are the nutrients that plant mostly need, like magnesium, then potassium, ammonia, calcium, ammonia especially for nitrogen source mainly. The micronutrients are cobalt, copper, iron, manganese, zinc. Okay, now obviously they are not found in elemental form, they are found in compound form, so we will mix these nutrients one by one. By first we weight them, what at what concentration, at what volume of medium we are preparing, then uh, according to quantity of that medium we will weight them properly in the weighing machine. Okay, digital weighing machine, and then we will mix them slowly in the water containing that appropriate volume. Okay, in a mild volume, and continuously it is shaking also, so we have to dissolve them slowly, and we have to use those chemicals first which dissolves more rapidly. Okay, then sucrose is also used. Okay, you can also use glucose also, but we use sucrose here as a carbon source. Vitamins like thiamine, pyridoxine and myoinositol is normally employed. For amino acids we use the glycine source, and for mostly lastly we use the hormones, the phytohormones at certain concentration, for the induction of roots, for the induction of shoots, and also for the induction of callus. pH has to be adjusted between 5.6 to 5.8. Now normally we all know that how we do adjust the pH, by providing the HCl, dilute HCl and dilute NaOH. Okay, few drops we dip them for maintaining that pH. Then finally we have to add agar. If you wish to prepare a semi solid medium, we have to add agar. Agar is given at a certain percentage. um, I think 8%, I think no, point 0.8 maybe so. And then we have to dissolve the agar. For dissolving the agar we have to use heater. For dissolvement of the agar we can also use your microwave oven also for heating that, melting that agar in that medium. And then we have to transfer that medium into the vessels, into the tubes where we want to inoculate the explants actually. And finally, together with the instruments, we have to autoclave them at 120 degrees Celsius with a pressure of 15 Pascal for 20 minutes. Now for the preparation of explant, what we do, so you can see so much of laborious technique is employed. That is the main fault of plant tissue culture: costly, one time investment, very costly and laborious, but the result is very good. We can prepare large number of plants within a short period of time. We can do commercialization. Okay, it's a good business also. For preparation of explants, the explants are taken from the meristematic zones preferred having high cell activity. That means from the meristematic zones, we normally use the explants that have a high cell activity, which is free from viral contaminants, free from the bacterial contaminants, and where no such type of the chromosomes are maintained at diploid level. Then transfer that explants immediately into a clean water, so that to avoid entrance of microbes, contaminants from the cut and exposed part, and to avoid browning due to phenolic oxidation. You have seen that when you cut certain plant, after some time that particular portion of the plant become brown, and that occurs due to phenolic oxidation. To prevent that phenolic oxidation, we have to immediately transfer that cut portion of the plant tissues into that water. Then we have to perform surface sterilization. For surface sterilization, first we have to cut the explant into little bit small pieces. Then we use a mild detergent of 2 in 20. Generally we use 2 in 20 as a mild detergent. And then wash in mild antibiotic, maybe fungicide or antibiotic solutions. And then repeatedly we have to wash them in double distilled water. Then comes inoculation of explant. How the explants is being inoculated into that medium. Okay, now for inoculation of the explant, those who are preparing to inoculate that explant in the tissue, in that culture vessel containing the media, sterile media, first we have to take air shower. Now this is an easy job. The air shower is very easy job. We have to take air, fresh air we have to take, and the clothes that we are wearing should be neat and clean. Clean lab coat should be used. Okay, not even that lab coat which has been used for the previous experiment. Clean lab coat should be required. At least for 20 minutes we have to take air shower. And then we have to, for sterilization of the laminar airflow, because previously some students have or some researchers have used that laminar airflow, so it has to be sterilized. For sterilization of laminar airflow, we have to switch on the UV light, taking care that we have to shut down that hood. Okay, that hood, so that the UV light does not come contact to your body, because it is dangerous. You have studied molecular biology previously, that how UV light causes mutation. For 30 minutes it has to be used. That will kill the microbes if present in the laminar air hood chamber. Then after that we have to put on the air switch and the chamber light. Then clean that whole area with 70% ethanol on the surface area. Okay, the explants are then washed repeatedly with now all the stops will be done within the laminar airflow. All the inoculation of the explants into the culture media will be done in the laminar airflow. So explants should be washed with mild mercury chloride. About 0.01% of mercury chloride is used for the last sterilization of the explant. The mercury chloride is extremely poisonous, so care has to be taken. High concentration should not be used, otherwise it will damage the whole tissue. And it is washed repeatedly. Then it is washed with double distilled water several times prior to inoculation. Then after that, the petri plate which contain the filter paper, that has to be used now. We have to place that explants into that filter papers within the petri plate so that the water can be absorbed, and we have to cut out the exposed portions of the explant which has directly exposed with the mercury chloride solution. We have to cut down that part little bit. Then we have to insert that explant into the culture medium vessel or culture medium tube with the help of sterilized forceps. All these things is done within the laminar airflow in front of your spirit lamp, right? Or you can also use bunsen burner with a low flame so that we can able to completely do the process aseptically. Okay, then label properly in the glass, the jar that we are using. We have to label properly at which date you are being you inoculate them with the name of the plant. Also you can give a code also that this type of plant is there. Okay, then incubation of the culture vessel. Normally the culture vessels are maintained in the culture lab. Conditions of 25 to 27 degrees Celsius are maintained. 40% relative humidity, although I have said 50%, 40% relative humidity is also quite satisfactory for maintenance of the culture. Then further we have to check them periodically, after 2 to 3 days repeatedly we have to check them that if there is contamination we have to side them. We have to remove them from the side from the other cultures, otherwise that contamination can also cause the other vessels as well. For subculture we have to again do the same procedure, but this time we will not do the surface sterilization of the explant, but the other things we have to do repeatedly for the subculture when we are transferring the uh, plant tissue in a new vessel containing certain changes in the concentration of the hormones, maybe for the induction of shoots or roots. Okay, now this picture is taken from the YouTube source where you can see that how aseptically this researcher is actually inoculating that explant into the culture tube in front of a laminar hair flow and having the spirit lamp. Generally we use spirit lamp, but you can also use Bunsen burner with a low flame. Okay, if a setup is available there, otherwise you can go for spirit lamp as well. Then after the culture of the tubes, then what we will do after the culture of the tubes, when we see enough growth of the plant has been occurred, we have to acclimatize that plantlets into the pots, because at the end of the thing the main objective of our thing is that we have to prepare many plants, okay, in a greenhouse. Okay, now this plantlet acclimatization is done in a greenhouse room. We have seen greenhouse chambers, large greenhouse chambers in respective farms, in nurseries, and they are subjected to different type of shocks as well, like temperature shock, humidity shock, nutrition shock, carbon dioxide level shock, and airflow shock. Now these shocks are actually being done so that to see that whether the plant can withstand in that conditions or not. Okay, now how do the plantlets are being subjected for acclimatization? Plantlets are placed in pots, small pots that contain garden soil, sand, and well decomposed farmyard manure in a 1:1:1 ratio. So we use garden soil, sand, and farmyard manure, which is actually prepared from your various kind of animal excreta with other materials like crop residue, then waste, vegetable waste, that prepares the farmyard manure. So the farmyard manure, garden soil, and sand is actually maintained at a 1:1:1 ratio, and in that composition and in that mixture we have to put that plantlets. Now this is the main time where we have to observe, because initially the plantlets were growing in such conditions where we are providing all the nutrients that a plant needs. now the plant has to absorb the nutrients from this mixture. That's why we say them acclimatization. This is the time where the plantlets will be subjected to this type of shock, that whether the plants are able to absorb the nutrients from the soil or not. Now, other medium mixtures are also being used like soilrite, vermiculite, perlite, coco peat can also be used in the potting mixture to grow these plantlets. Okay. After 45 to 60 days, these acclimatized plantlets are shifted to the nursery, and from that nursery, we bring the plants, or if it's a big nursery, we can go for commercialization. If in case of monocotyledonous crops, right? So, this is the whole process of your the plant tissue culture. I hope you have able to understood this part. If you have any doubts, you can reply in the WhatsApp group, and in the next class I will try to discuss about the other things that are actually given in your plant biotechnology syllabus.